Assalamu alaikum. So here we'll be talking about something that is really exciting. This is sort of like the future of molecular biology and the future of medical treatment, gene editing, changing genes. And we'll talk about the how it goes, how it was discovered, how it works, and we'll talk about potential aspects of this technique. CRISPR Cas9. I'm sure you've heard it on the news. I hope you have. If you haven't, then I don't know where you live, Mars or something. You're with the James Webb Telescope or something. I don't know. Read about it. So let's start. Okay, so what is CRISPR Cas9? CRISPR is an abbreviation for clustered, regularly interspaced, short palindromic repeats. What do we mean by that? So we talked about repeats. So basically, we're talking about sequences that are repeated. That's one. Two, they are palindromic, mutanadira, meaning that you can read them from one strand, and on the other strand you read them exactly 5' to 3' the same exact way, just like restriction sites, which we talked about before in the first lecture and in the previous course as well. Okay, then these sequences, these repeats, are short and they are interspaced, so there are spaces between them, and these spaced sequences, repeats, they are regularly interspaced. That is, there are specific distances between one repeat and the other. And they're clustered, meaning that they're all located as clusters, as groups of sequences. Okay? And where does that exist? It exists in bacteria. So what you have in here is that you have these black boxes are the repeats, the palindromic repeats. Okay? And they are separated by other sequences, and these sequences are viral. Okay? So this genetic system exists in bacteria, and it is sort of like the immune system of bacteria against bacterial phages. Now, Cas9 is a nuclease. It is an enzyme that cleaves nucleic acids, and it is RNA guided, so it does not cleave anywhere. Rather, there is an RNA molecule that is associated with it. Okay, so the enzyme is a ribonuclease. You know how we talked about proteins, and we say glycoproteins, that is proteins that are associated with sugars, lipoproteins, proteins that are associated with lipids. We have riboproteins and we have riboenzymes. This one here is a ribonuclease. So it is an enzyme, a protein enzyme that is associated with RNA molecule, and this RNA molecule takes the nuclease to certain sequences. So it is RNA guided nuclease. Now this enzyme right here can make single or double single strand breaks or double strand breaks. So it creates NICs, N-I-C-K-S, plural. Okay, so that's what it is. This is the nuclease. So it's a protein right here, and it is associated with an RNA molecule, and it's called the guide RNA, gRNA, guide RNA. And this is the genetic system, that is the CRISPR. Great. So what is the biological function of the CRISPR-Cas9 system? What is its natural function? I said that it functions like an immune system. It is the immune system of bacteria against bacteriophages or phages, the bacterial viruses that we talked about before. So basically what happens is the following. When you have a bacteriophage infecting a bacterial cell, it sits on the cell membrane, and it inserts its DNA inside bacterial cells. Now what happens is that this DNA integrates into the bacterial chromosome, and then it gets transcribed, translated, and it hijacks the bacterial cell itself. So bacterial cells have been created in a way, or they have evolved in a way to invent a system to protect themselves from bacteriophages. They created, or they had developed restriction nucleases, and we talked about those earlier. Now, the CRISPR-Cas system is something similar. So what happens is that it's a sophisticated system. So what happens is that you have the bacteriophage DNA now inside the bacterial cell. Now, what the bacterial cell does is that it activates enzymes like restriction nucleases, and these enzymes cleave the bacteriophage DNA. The bacteria then take this piece of DNA and it inserts it into its chromosome. So you have, in this cluster, you have several sequences, different sequences taken from different bacteriophages that previously infected this same particular bacterial cell. All right, and these different bacteriophage DNA sequences are separated by these palindromic sequences. So this is the CRISPR system, clustered, regularly interspaced palindromic repeats. So you have these are the repeats in black, and these in color are the pieces, the graded pieces of bacteriophage DNA. And this system, this genetic system, can be transcribed, producing several RNA molecule, and these are known as the CRISPR RNA. And then these are cleaved into different RNA molecules. Okay, so you have the RNA molecule containing the bacteriophage, the transcribed part of the bacteriophage DNA, plus the bacterial DNA as well. Okay, the repeat, and note it's palindromic, and that's why it gets hydrogen bonded. You have the stem loop structure. Okay, great. Now, when you have another bacteriophage infecting the same bacterial cell, that is the same bacteriophage that previously infected the bacterial cell, infecting the same bacterial cell, what happens is that you have this piece right here of RNA that integrates, it binds to the Cas9 enzyme, and it guides the enzyme to the bacteriophage DNA. And the Cas9, what it does is that it degrades the bacteriophage DNA so that it does not infect the bacterial cell. Beautiful, sophisticated system. And this is similar to how our immune system learns of previous infections or exposure to antigens. Now, years ago, a Portuguese scientist known as Francis Mojica discovered the CRISPR system in bacteria. And he studied the biology of this system. Okay, now, and he's a modest man. Anyhow, so years later, these two scientists, a French scientist and an American scientist, took advantage of this system. And this is the beauty of science. It's taking advantage of normal biological systems. And they got the Nobel Prize in 2020. After their few years after, they published their work. Geniuses, female power. Now, no one remembers Francis Mojica. I read about him years ago, but I decided to dig in to find more about him. Or actually, I didn't read the article; just got his name. So remember, modest men do not make history. You need to do something really good and brag about it if you want. Now, these two scientists took advantage of this system and they said, why didn't we do gene editing using the CRISPR-Cas9 system? And that's what they did. We previously talked about DNA repair mechanisms in the molecular biology course. And I told you that there are several systems, and we briefly talked about these two systems, the homology-directed repair and the non-homology and joining repair systems. And these two systems are activated whenever you have DNA breaks. So whenever you have this DNA break, and at different locations, for example, if the DNA is hit by an X-ray or something like that and it's damaged, the cell takes advantage of the presence of a homologous DNA. Remember, we have two DNA molecules. We are diploid. Our cells are diploid. We have one from our father, another from our mother. So if one chromosome, one DNA is damaged by breaking it, well, you have the other chromosome, and this system, the homology-directed repair, sort of like does exchange process whereby it takes the part of one strand of the good chromosome and it puts it on the other chromosome, and then you have filling in the gap taking place. And the other system is whenever you have real damage in the DNA and it's non-homology, so it does not really depend on the presence of a homologous chromosome. Rather, what you have in here is something like a glue whereby these two strands are glued together. So just like when you break a vase or something like that and then you glue the different pieces, well, you can see that it's damaged, right? I mean, yeah, you can see that. So anyhow, so if you have the two pieces of DNA glued, that results in creating an indel. What does that mean? It means indel means insertion, deletion. Okay, so you have indel mutation. So just like a vase, you can see that it's fixed. It's not like before. Well, same thing with DNA. You have either insertion of nucleotides or deletion of nucleotides as the cell glues the DNA strands together. And that results in the creation of a defective gene. So the gene is defective, it doesn't function, it's not normal, it's mutated, it doesn't function anymore at all, or doesn't function properly. So it's like knocking out a gene. It's like removing a gene from the human cell or the human genome. Now the other system, the homology-directed repair system, where you have taking a piece of one strand from the good chromosome and it puts it on the other chromosome, well, this is something like you're correcting a mutation. It's almost like you're replacing a piece of the chromosome. It's like you can insert a fragment of interest into the chromosome, and that's exactly what they did. So here's the idea. To replace a gene in a human genome, the homology-directed repair system is activated. And it's activated by using a Cas9 gene that can produce a protein, a nuclease, Cas9 nuclease, that creates a single cut, not two cuts, just a single cut. And that would activate the homology-directed repair system. Now, how does Cas9 do that? Well, recombinant DNA technology, you have the insertion of the Cas9 gene into a plasmid, along with the RNA molecule that would be integrated into the Cas9 protein. So it's recombinant gene, a Cas9 recombinant gene. Okay, so this plasmid is introduced into cells, And then, since the RNA is now part of the Cas9 protein, the Cas9 protein is directed by this RNA to a region in the human chromosome that is complementary to this RNA. And the RNA is complementary to a gene of interest, the gene that we want to replace. Now, along with the Cas9, with the recombinant Cas9 protein or gene, you have the introduction of the gene or the part of the DNA that we want to recombine into the chromosome. That is the part of the DNA to replace the gene that we are targeting. So we are fooling the cell. We're telling the cell, oh, sorry, we just damaged your DNA. Well, here's a replacement. You can take it. So forget about the homologous chromosome. Here's one for you. So what the cell does when the homology-directed repair system is activated, you have the Cas9 taken to the chromosome that we want to damage. It damages the chromosome at a certain place, and you have the activation of the homology-directed repair system. And then the system, or that is the homology-directed repair system, uses the fragment that we are introducing as well as the DNA to replace the damaged DNA. Okay? And this is the idea behind it. Genius stuff. So again, here's what happens. We introduce the Cas9 gene with the guide RNA. The RNA that would guide the Cas9 to the region of DNA that we want to damage or replace. So you have the Cas9 guided by the RNA to the region of interest in our chromosome, or whatever chromosome, mouse, bacteria, human, whatever. And the Cas9 creates cuts. Now you have activation of the homology-directed repair. So that this system, remember, needs a DNA to replace the damaged DNA, and we introduce to cells this DNA. So we're again fooling cells. We're telling cells, well, why don't you take this instead of using the homologous chromosome? So what the system does is that it uses the system that we are introducing, and it replaces the damaged DNA. So we can have let's say a defective gene that exists in cells. We introduce the normal gene, right? So we're replacing a defective gene with a good gene. We're fixing things. Or we can do the opposite. We want to study a certain gene and we want to see what would happen if this gene becomes defective. So we take cells, we introduce the Cas9 system with the DNA that is defective. And cells take the normal gene out, the good gene out, and it puts in the defective gene inside the chromosome, and then we can study what this gene does exactly and why it's important. Now, the Cas9 system can again cleave DNA, and it can activate, and cells would activate the non-homologous end joining system. And what cells do is that they take DNA and they glue it. And that results in the creation of indel mutations, insertion deletion mutations. And these insertion deletion mutations would lead to frame shift mutations, right? That is, you have you're disrupting a gene. You are damaging a gene this way. So let's say that we have a gene that causes cancer. Well, we can just damage it. Normal cells would not be greatly affected. but cancer cells would. What if we want to study a certain gene? Well, let's ruin it, let's disrupt it, let's mutate it. So we can use the Cas9 system to mutate this gene. So there are so many things that we can do with the gene and with this system, the CRISPR Cas9 system, and that's gene editing. We are changing genes, we are editing genes. There are other uses for the CRISPR-Cas9 system. Someone might say, well, why don't we use the Cas9 system to guide certain proteins like transcriptional factors to a gene, to activate it or to repress it? So they would have the Cas9 protein associated with the guide RNA. But the Cas9 does not function, it's defective, okay? So we do genetic engineering, we mutate the Cas9 protein so that it goes to the region of DNA of interest, but it does not cut the DNA. And we have an activator, a transcriptional activator associated with the Cas9 system. So we take the activator to the gene that we want to activate. Or we can do the opposite. We have a repressor, a transcriptional repressor associated with the Cas9 system. So it goes, let's say, to the promoter proximal element or to the promoter region or to the silencer in a certain location in a chromosome, and the repressor would suppress transcription of a certain gene. Someone might say, well, why don't we have a green fluorescent protein associated with the Cas9 system? We want to see what cells take up the Cas9 protein or the plasmid that contains the Cas9. We want to study these cells and we'll have individually, and we want to see what happens to them if we do this or that. So you can see in here, for example, these are cells, okay, under the microscope, the light microscope, and these are the same cells, and the ones that fluoresce, that give this green color, are the ones that have the Cas9 system, or the Cas9 protein. So we can study, specific cells as well. Since the CRISPR-Cas9 system has been discovered, we've had the discovery of other Cas enzymes and proteins. An example, and other systems as well. An example is a Cas enzyme known as Cas12a. And this system introduces staggered cuts rather than blunt cuts. So the Cas9 enzyme creates blunt cuts. On the other hand, the Cas12a system creates staggered cuts instead. You have the discovery of another Cas enzyme known as the Cas13a, and this enzyme is specific for RNA endonucleases. So there are different enzymes that have been discovered, and scientists are trying to be creative about how to use these enzymes. I probably showed you these images before, but this is what scientists are trying to do. They're trying to use our fingerprints in certain ways. They're trying to edit DNA. They are trying to change DNA using the CRISPR-Cas9 system. They try to cut certain regions either to fix them or to disrupt these genes. It's like using the CRISPR-Cas9 system as a tool, wrenches and bolts, trying to fix DNA. And the future really is something like this. Instead of taking a pill of chemicals, we're going to be taking a pill of DNA, the CRISPR Cas9 system probably, or something else, something similar. Now you can go back to newspapers and talk about and read about what scientists are trying to do. You can Google it. there are so many things that scientists are trying to do. Some of them are outrageous, like trying to genetically modify human embryos, like the scientists right here, again trying to use the CRISPR Cas9 system. You can read about it in the news, but there are so many things that scientists are trying to do. Some of them are really, really cool. Some of them are really earth-shattering, really great in trying to fix our DNA and treat diseases. But bad things happen as well. You can read about this as well on the net. This Chinese scientist modified the DNA of twin sisters. Um, I don't know, trying to sort of like he disrupted a gene, a certain gene, and this gene is called CCR5. This gene is important for the entry of the HIV virus into our cells. So by disrupting this gene, these girls are protected against HIV. However, this gene is also important, seems to be important for intelligence. And we don't know what would happen to these girls. And what he did was so unethical. He was imprisoned actually for 3 years by the Chinese authorities. So, bad things happen. Now, and this leads me to the sort of like the final say. What scientists are trying to do is really cool, except that it can be used for bioterrorism as well. There are certain things that can be done, including modifying the human genome. Okay, modifying the human microbiome as well, modifying bacterial genome, pathogenic bacteria, making existing bacteria more dangerous, making biochemicals via in situ synthesis inside our cells, recreating pathogenic viruses. Okay, so again, bad things happen. You need to be, your eyes need to be wide open. You need to be smart about how good things can turn into bad things. So um, you have this chart right here that shows different categories of bioterrorism. So you have category A is the most dangerous. You have modified anthrax or smallpox, but you have not so dangerous pathogens as well, such as the HIV and influenza. Well, it is, it is, of course, HIV is bad. But it wouldn't spread, widely like anthrax or smallpox. Okay, so